Hello everyone, my name is Nikola Stankovic. I work at the Faculty of Sciences and Mathematics, University of Niš in Serbia. But during 2023 and 24, I was on my postdoc at the IGB Institute in Berlin, Germany. And there I was involved in one very interesting project, Platon Divatox, where we were investigating the association of toxic cyanobacteria and macrophytes in Berlin lakes. The topic of this presentation is spatial and seasonal patterns in macrophyte-associated toxic cyanobacteria and their toxins. So that was a case study from a temperate lake. Objectives of this research were to investigate the presence and distribution of macrophyte-associated cyanobacteria. So during this research we collected material from several Berlin lakes, but we were mostly focused on Lake Tegel or Teglase. The other objective was to quantify neurotoxins, anatoxins concentrations in macrophyte biofilms, but also we were using microscopy, cultivation, and molecular analysis. Then one of the objectives was to examine ecological factors influencing the occurrence of anatoxin-producing cyanobacteria, parameters like Secchi depth, oxygen, phosphorus, sulfate, as well as cyanobacterial community composition. Then also to evaluate the risk of macrophytes as vectors for cyanotoxin transport, and also finally to contribute to ecosystem management strategies. So on next slide you can see locations where we collected material for analysis, and you can see the part of on this map part of this lake. We collected material from 4 different transects, T1 to T4, but also 2 additional locations, E1 and E2, and here on this map is labeled, shown where we collected from E2 location. We collected the most toxic samples. On this slide you can see methodology how we collected samples. So let's say from each transect we collected in total 16 samples per month, 4 replicas per each meter of depth. For collecting material we were using rakes with telescopic sticks, and also water sampler for collecting water samples from specific depths, of course. And on this slide you can see how we prepared our water samples for further analysis. So one part of these water samples we were using for detaching cyanobacterial biofilms and let's say cyanobacteria from the macrophytes, and after that we were preparing samples for molecular analysis, or let's say for DNA extraction. For one part of these water samples we were using for quantification of toxins, and also we were using microscopy, of course, to find and to quantify cyanobacteria. And also one part of these water samples we were preparing for chemical analysis in chemical lab, of course. And what we found is that the most toxic samples contained trifle cyanobacteria from the order Oscillatoriales, with the most common being cyanobacteria from the genera Microcoleus and Oscillatoria. And by using molecular methods and the results from this analysis, we found positive correlation of the presence of anatoxin A synthetase in our samples, and positive correlation with Microcoleus that was present in these samples. Also, we were working on we were trying to find a link between, of course, the presence of Microcoleus, so let's say the abundance of Microcoleus, with the positive samples on PCR for this gene. And of course we have a huge amount of results. We try to find, are there any patterns in depth, that actually is there any influence of depth and on presence of Microcoleus, and let's say is there any influence on toxicity. Also we were working on the seasonal distribution to find is there any specific period of the year when we can collect, when we can expect actually these toxic samples. But the most interesting results that we actually found is that we found positive correlation between concentration of anatoxin A and the abundance of microcolloids, and also concentration of dihydroanatoxin with the presence, or let's say abundance, of microcolloids, and also positive correlation between dihydroanatoxin and anatoxin A in our samples. And I hope we will publish these results by the end of this year. Many thanks for your attention.